This is a presentation on crystallography. As it may sound, crystallography is the study of structures. In the science world, it is primarily used to study proteins and other biological molecules that don't crystallize naturally. It is also used to determine the atomic and molecular structure of a crystal. By measuring the angles and intensities of the diffracted beams, a crystallographer can produce a 3D picture of the density of electrons within the crystal. From this electron density image, the mean positions of the atoms in the crystal can be determined, as well as their chemical bonds, their disorder, and various other information. It has helped reveal the structure and function of many biological molecules such as vitamins, drugs, proteins, and nucleic acids. How does crystallography work? So to begin with, there are three types of beams: X-ray, electron, or neutron. X-ray being the most commonly used in the science field. Crystallography methods analyze diffraction patterns of a substance caused by shining a beam of radiation to it. That is what being showed in the first image on the top left corner. So first, a stable crystal must be created. To form the crystal, the molecule is held in a solution and along with a buffer and precipitants is sealed within a reservoir. Once a stable crystal has been formed, it is introduced to the crystallography equipment, then a beam is directed at it. Synchrones are the most common source for the beams, and they also produce the most complete diffraction patterns. So this is seen on the second image to the right. You can see the crystal after it's been shined with the X-ray. You can see the diffraction pattern, and then after that you can see what a crystallographer might do with that diffraction pattern. They might do an electron density map, and they can see the atomic metal of the crystal. With any methods, there are some limitations. To begin with, the sample must be crystallized to begin with, so this obviously limits the sample. Second of all, membrane proteins and large molecules are difficult to crystallize due to large molecular weight and poor solubility, so this also limits the sample that can be used. Third one is minimizing background noise. The diffraction patterns produced by X-rays passing through a crystal contain signal and noise, and separating the two can be very challenging. The noise can lead to inaccuracies in determining the structure, and it can also make data analysis more complex. Lastly, cost and accessibility. Equipment is costly, and it may take several attempts to crystallize a protein, so it might not even be on the first attempt. Let's compare X-ray crystallography and NMR spectroscopy. Some of the advantages that X-ray crystallography has over NMR spectroscopy are the following. It provides a two-dimensional view that gives an indication of the three-dimensional structure. It is relatively cheap and simple compared to NMR. It is useful for large structures. It can yield high atomic resolution, and it is suitable for water-soluble proteins. Both of these techniques are used to study atomic structures, but these are just some of the advantages that X-ray crystallography has. Some of the controls needed for crystallography are temperature and a humid-controlled refrigerator or room for crystallization of the plate and incubation, protein at concentration of 300 to 600 micromolars, a 96 well sitting drop crystallization plate, and one X-ray generator and two detectors. In this first research example, researchers were looking at electron diffraction patterns of crystals. So in this specific study, they were using electron crystallography. The researchers were able to look at the diffraction pattern and assume crystal size by the pattern of diffraction. So in the first column in the image, you can see the crystal suspended in the solution. In the second column, you can see the diffraction pattern that was seen, and the circle drawn around it were the researchers finding out the crystal size from the diffraction pattern. In this second research example, the researchers were looking at different geometric positions of the crystal and their effects on the X-ray diffraction results. They were looking at the crystal delivery by modifying the microfluidic chip that rotates the crystal. Specifically, they were looking at lysosomal crystal structures. So in the first row is the first crystal position, and then the position is moved by a degree of 10. The second row is after the microfluidic chip is moved to a different position. So it's the same experiment before and after the microfluidic chip was moved. The image at the bottom shows a lysosomal structure after structure determination. So that's how it's seen after they've looked at the diffraction pattern and created this image. For this third research example, it is important to know that X-ray diffraction becomes a bit complicating when attempting to measure interatomic distances for metal complexes such as lithionine and actinide elements. So the researchers in this experiment found a method that makes it possible to diffract metals. All in all, they found that having a calyx as a scaffold to crystallize the small metal complexes helps in the diffraction pattern. So in the image to the right, you can see the steps it takes to perform the X-ray crystallography. I think that shows a very good example of that. On the image to the left, you can see how ceracolin, which is a protein that they use in the experiment, and how it binds to a wide range of metal complexes. It shows the structure of the protein bound to iron and other metals. Just to tie it into class, we went over different X-ray sources and methods that utilized X-ray. So this would just be an added-on method that also utilizes X-ray. Another one is we spoke about electron scattering. This is pretty similar to what happens in X-ray crystallography with the diffraction and the beams.